Hello everyone. Today I am going to talk about the experiment used to extract DNA from fruits such as strawberries or bananas. The aim of this experiment is to extract DNA from fruit so that it shows DNA is present in living cells. And the common fruit we use are strawberries or bananas. To ensure the experiment is safe, safety goggles should be worn, and care must be taken when handling chemicals and glass equipment. The reason for choosing fruit for the practical is that fruit is a good source of DNA. Its cells are soft, easy to break open, and contain a large amount of DNA. Strawberries are often used because they are octoploid, meaning they have many copies of each chromosome, making the DNA easier to be seen. The first step of the experiment is to break down the cell walls and cell membranes. The fruit is mashed or blended to break up the tissue and increase the surface area. This releases the cells so that the DNA inside them can be extracted. Next, detergent is added. Detergent breaks down the cell membranes and the nuclear membranes, which are made up of phospholipid, allowing the DNA to be released into the solution. The detergent also helps separate the DNA from other cell components. Salt is then added to the mixture. The salt helps the DNA strands clump together by neutralizing the negative charges on the DNA backbone. This makes the DNA easier to separate from proteins in the cell. The mixture is then filtered using filter paper or a sieve to remove solid pieces of fruit. These leave a clear liquid containing dissolved DNA and other cell contents. After filtering, cold ethanol or alcohol is carefully poured down the side of the container. DNA is insoluble in alcohol, so it separates out of the solution and forms a white, cloudy precipitate. The DNA can often be seen as stringy strands floating in the alcohol layer. The DNA can be gently collected using a glass rod or wooden stick. This DNA is not pure, but it is visible to the naked eye and can be used to demonstrate that DNA is present in living cells. In conclusion, DNA extraction from fruit involves breaking down cell walls, dissolving membranes with detergent, using salt to help DNA clump together, and alcohol to make the DNA visible. This practical demonstrates that DNA is found in living organisms like fruit. I hope you enjoyed this video. Thanks for watching.